So thank you to be here and to the organizer for inviting me. I wouldn't say it's very nice to come to Paris because I'm just next door. But, okay, so I will, somehow this is a sequel to Hugues' talk, but it will be especially about cell motion and collective cell motion. And so this is some work that has been done in collaboration. Is very nice with the chairman. So this is with Pascal and his student Laurence Petitjean, Olivier Cochet, and Maxime de Forêt. And the theoretical part has been done also with Nestor Sepulveda, who is now in Chile, back in Chile. Okay, so I'm also a bit impressed because it's people say it will be on YouTube, this lecture, but then Hervé just told me that the best way to be famous is just to say silly things, so that's perfect. Okay, so cell move collectively. So I will start by giving you some example just to motivate the talk and why we are interested. Not in random motion of sheep, birds, things like that, but we will focus specifically on cell and try to show you that this is an interesting topic. Of course, you're welcome to ask questions at any time. So this is the motion. This is a fish larva, zebrafish larva. This is a small group of cells that moves along the lateral line of the fish. And this group of cells is moving on a track that has been characterized chemically. And it deposits small clumps of cells in the back, which are going to do this mechanosensory organ of the fish, so some peripheral nervous system organs. And so what you see is that all these cells, they move together. So they form, and I'll show you on the next slide how it is organized. Oops. Okay, so this is the tip. So there are some actually leader cells that guide this thing. And all these cells move together, and you'll see that in the back at some point things will detach. And I forgot to say that these movies are taken in Darren Gilmore lab at EMBL. So this kind of motion where cells move together, so classically there are two kinds of cells, so cells that are epithelial, if you want, and then they undergo this epithelial mesenchymal transition where there are single cells that move and do things. Sometimes they do bad things when they are tumor cells. But so now more and more it has been realized that cells can also not only move as single cell but move together, and that's the case in many steps in biology. So of course one of these is pathology. So the people are interested in, as I said, in this collective motion because this has been particularly stressed by Peter Friedel, that there is an analogy between this, say, organized motion, for example, in the fish, and some cancer, some carcinoma cell that invade as a group tissue. So here you have some analogy between what I just showed you, this lateral line primordium, and invasion of a tissue in some mammary cancer, carcinoma. So that's one thing, but there are many other examples that are both in development and other kind of things. So one example that has been, I think, is a very nice study in Roberto Meyer group in London is the motion of neural crest cell. So neural crest cell. So this is a poster that I just took from. Eric Thévenot and Roberto Meyer in the development recent that appeared last year. So what are these neural crests? So these neural crests are just they just appear on the side of the neural plate. So there is a neurulation here, and then this neural class cell. And then all along the embryo, they move like this and they avoid some obstacles. So they move in streams and then differentiate into a number of very different cell types here. And this motion has been studied quite a lot. So this was a very nice model, and you can read about that. And so that's another example that is been studied a lot is Drosophila, and particularly in the group of Denise Montel, Drosophila oogenesis. So Drosophila oogenesis here there is a small reminder of how it is. So you have the ovary and then this ovariole, where you have the maturation of these eggs in beads. So the first stage are at the tip here, and then more, and then they progress as they mature, and they are linked between these. There are small link between these. So if you take a picture like here, at a given moment, you see eggs at different stage. And so in this egg, there is an important motion, is the motion of border cell, which move here. So like this, they move along this egg, and this has been studied. This is a group of cells that moves in this egg as a group. And it's very important because it moves to deposit some RNA that bicoid RNA in the egg. So it moves like this here, and it will deposit bicoid RNA that will make the gradient that will give anterior-posterior positional information in the embryo, in the Drosophila embryo. So that's one group of things. And there are also things that are more exotic that were discovered in the group of Builder that I will show you now. Oops, this is not very well. Okay, so this is that these eggs, in fact, at some stage, between here, stage 7 or 8, they rotate globally. Okay, that was a very big surprise for people because people have studied this for quite a while and never realized that the egg could rotate like this globally. And so this global rotation of the eggs is actually global movement of the cells that are on the surface of the egg that are moving together on the extracellular matrix. So this gives, they organize their motion, and presumably there is a coupling between the motion of the egg, of the cells and the extracellular matrix, and they organize the extracellular matrix. And the idea is that it makes the extracellular matrix doing like what people call a corset, so that then the egg will elongate, and so it will be much more difficult to elongate in this direction than in this direction. So it will go from a round shape to some kind of elongated shape. So that's.
Excuse me.
Yes.
The red bodies are your stem cells?
So the red body are, so if you have are the nurse cell. So if you look at the egg of Drosophila, it's made of 16 cells here in the interior. One of them will give the egg proper, and the other one are the nurse cell. At a later stage, this 15 cell will undergo apoptosis, and their cytoplasm will fill this part, and it will elongate. It will give the egg like this. And on the top is what people call follicle cell that you see in green here. So you have 16 big nurse cell and 15 and then on top you have follicle cell. And actually this is a nice process. I mean, people are studying different kind of things for it. So in this follicle epithelium at a slightly later stage, there will be appendage formation, and that's also something that people have study this, how you buckle this epithelium to make a small tube.
So is there a preponderance of
Is there what?
nerve cells? Is there like a priority for nerve cells to form before other kind of cells do get formed? Something like that?
So I think I'm not so sure.
Have like differentiation for
No, so you see here, so you have the germanium, so you have the follicle cell here, and then you have this division of nerve cell, and then the follicle cell also cover the egg. But I mean, now you are touching the limit of my knowledge, I should say. So, okay, so let's proceed a little bit. So now, okay, so that was in some sense normal development. But of course, there is also cell motion when in pathology, and one of them is wound healing. So that's a process that is important, that is actually very complicated in the real case where you have a wound, because there is many steps. First of all, you injure different layers. There is inflammation. You have to react a lot to prevent bad things to enter the body. Then you have to really seal these things, so there is proliferation, and then migration, then maturation. And I mean, this is a process that people would like also to understand to prevent. So one bad thing with wound healing is that in some cases you have big scars and you want to avoid them. So if you understand a bit more this process, you can hope to heal without scars. So this is a very interesting process, but quite complicated. So for a long time, people have studied models of wound where you scratch some cells in vitro in Petri dish, and Pascal, I'll show you the Pascal version of that, which is a bit even more model, but more reproducible. So the idea is to use microfabrication technique to have a wound where cells are not, I mean, a very clean wound. So this is the microfabrication thing, and then you see the cells here, and then you remove this stencil so that you have sheets of cell in vitro, and then some space between them that is this kind of model wound. But the nice thing here, I mean, if you want at least to have reproducible experiment, is that this is very clean, and so the cells are in good shape, and then you can study the motion of these cells. So that's a setup that I'm going to describe in the following. I just show you a movie from Pascal, showing you what is going on here. So first of all, when you remove this stencil, you induce motion in the bulk here. The cells start to move, and then on the border with free surface, you see that there is invasion, and invasion is non-trivial. I mean, there are large protrusion. Oops. So maybe I take another. Let's change this. Okay. So there are large protrusion, and there are some cells that are different on the tip of this protrusion. So there is collective motion, but also some kind of leader cell on tip of this finger that invade here.
What is the media they're moving in?
What?
What is the media they're moving in?
So they are not really moving on media. They are moving on glass. They are crawling cells.
And there's no extracellular matrix polymers?
So on this experiment, no. But some people do it with covering glass with fibronectin or some other substance.
So
And actually, but yes.
how do they move?
How do they move? We'll come to that, how cell move. So cell crawl, I'll show you how cell crawl.
It's somehow related to the last question.
No. So cell division is not, maybe it's part of it, but it's, so the total time is 15 hours. So the division time of a cell in this experiment, 20 hours. It doesn't mean that there is no division, because there are many cells, so there are still division. But it's not thought that that division is the main motor of this. And actually, you can put some drug to prevent division, like mitomycin, and then you still have this thing. Other question? Yes?
Why are some cells brighter than others?
What?
Why are some cells brighter than others?
What are some cells brighter? So this one, I think they just are not really as cells that are dying, that are just going above the epithelium. So these are not really interesting cells. They are bright, but not so interesting. Okay. And I just show you this leader cell just for fun. I mean, these are, so you have all these cells that are in principle identical, and one of them, for some mysterious reason, acquires a different phenotype. So it's much bigger. It has a big lamellipodium. We'll come back to what the lamellipodium is, but it allows the cell to crawl. And the blue here are the nucleus, just for those. Okay, so there is a different cell on the top, and very often, actually, it has two nuclei. When division appears, it doesn't succeed completely, and there are two nuclei. Okay?
Is the green label microtubules or the cytosol?
I think it's more actin. I think it's GFP actin. So microtubule, we won't speak too much about microtubule there. Here. So okay. So the nice thing about this in vitro experiment, which has of course easier, that cells are easier to visualize that in vivo, that in vivo experiments. So you have a lot of data. So for example, here with particle image velocimetry. Particle image velocimetry is a technique that is coming from fluid mechanics, where you just take a small bit of the image and you correlate it to the next image, and you adjust the thing so that it matched the best, so you can see how things move. And so for this, then you get the velocity field in everywhere in the picture. So it's like, if you want fluid mechanics, from this data, what you know is the velocity every 15 minutes, say, everywhere. Okay? And so you can have all kind of quantity, like the statistical distribution of velocity, the correlation between velocity at equal time at different times. So it's like a statistical mechanics problem, and one wonders what describes this data. Okay? So just because I just remind you about single cell motion. So I show you, first of all, the single cell motion. So this is a keratocyte, some kind of epithelial cell that moves. So it has this big lamellipodium that is useful to propel the cell everywhere. So you see that it moves quite nicely with this. Okay, so that's a keratocyte with lamellipodium. And there are other kind of motion that are a bit less efficient, as you'll see. So this is a crawling cell. The principle, basic principle is the same. I'll just remind you before, but here it's less, it's organized a little bit differently, where you have deep pseudopod that crawl, and you have also retraction. As in the other case, you have retraction in the back, and the nucleus is carried by this, this thing here. Okay? So I think, okay, so that was taken. So just to remind you very quickly what is cell motion, of course it will not, it's just a very basic summary. So what you have is you have actin here in the front of the cell. And so it's a complicated process where you have a mesh of actin, which is polarizing at the front and depolarizing at the back. So you have some kind of global treadmilling of this network of actin. And so this is pushes the front on the medium. And you have on the back of the cell, and you have adhesion, of course. And on the back of the cell, you have some contraction to pull off the cell. So you have some kind of motor here, and here you should take this out. And so this is a process that has been studied in quite detail, that is currently studied a lot. So there are many interesting questions. Of course, how do you really make this treadmilling, which is quite complicated with many different proteins, where you have many proteins that are implicated in this actin treadmilling. And in the back, you have also, you have this contraction where you should, you have contraction like this to remove the cell from the substrate. And you have, so there are a question of, first of all, the establishment of polarities. So you probably heard of it by Erwin Frey. So there are two systems that are of Rho GTPase that are inhibiting each other. It's thought to inhibit each other, so that you have a front and the back. And so there are interesting questions of how this thing propels, and also what is the origin of stochasticity. So you have seen that this cell, in fact, move, but move as a bit stochastic, stochastically. So how is this is generated is not really completely understood at the moment. So there are some, there are a lot of interesting questions. I will not touch upon them because of time, but so there are already at the single cell level a lot of questions. Okay, so now, as Hugues said, okay, there are all these kind of things, but for a long time, starting by Fürth in the 20s, people have tried to describe cell motion in a reduced way. Just because if you are interested maybe at a larger scale, you want to go back to the biochemical detail because they will influence what you see, but maybe you don't need to describe all this biochemical detail to start with. So the description that was proposed already in the 20s and then was studied a lot also in the 70s is just as Hugues was using for the fish, this Ornstein-Uhlenbeck process. So actually this was invented at the same time as this was used by Ornstein. So here what you have is you have a white noise. Okay, see, this is the velocity. So the cell will be reduced to a single point. And there will be some persistence given by the inverse of alpha, time. Alpha as the dimension of inverse time. So it will say that the cell persists in its motion for a certain time. And then there is a noise so that the cell will move randomly. So I just show you result in the 70s. So if you, this was motivated by looking at the mean square displacement. You know that if you have Brownian motion, this would be like going like with time. And here you see that at the beginning there is this time t star, which is the inverse of alpha. And so it starts smoothly if you look at the mean square displacement from a point after time t. And so this here just to say that it was done in the exponential growing phase when the cells do not see each other. So if you look at single cell, they basically follow this Ornstein-Uhlenbeck. So at least this is a very common classical description. So it was actually studied in much more detail in this paper by Flügberg group in 2005. So they came back and decided to look very carefully at the motion of cells. So what they did is that they followed cell individually and try just to, from the motion of cell from the individual motion of cell to deduce what a precise description of the motion. So for example, here they look at, so they look at their cell. Okay, there is this, the speed here, v, and if you have the previous description, what you see is that if you fix v, you expect and you measure the acceleration, you expect it to be Gauss distributed along the mean will be alpha v and minus alpha v, and then it will be Gaussian distributed around it. So they looked at that. So this is speed, and then you look at the acceleration both in the transverse direction and in the parallel direction. So in the parallel direction, you expect it to be in the mean minus alpha v. So that's exactly what you see. If you look in the transverse direction, you expect the mean to be 0. So that's also what you see here. But if you add this really description by Ornstein-Uhlenbeck, you would expect that the mean here is independent of the velocity. But actually, if you look at the data carefully, you see that the root mean square, actually the root mean square of both the parallel component and the perpendicular component and the parallel component are equal, but they increase with velocity. So this is S just because I don't know, my PowerPoint doesn't like Greek, but this should be sigma v and sigma 0 plus sigma 1. Okay, so this Ornstein-Uhlenbeck is a bit more complicated. The noise here depends on the v, okay, in principle for these cells. And also, if you look now at velocity autocorrelation in the Ornstein-Uhlenbeck case, you would have a single exponential. So if you look at the mean of vt prime, it would be exponential in the difference. And in fact, when in this data, you see that it's a bit more complicated. You have two exponential. And this, they can account very well with an exponential kernel here. So this would be some kind of refined model of this Ornstein-Uhlenbeck type, where you have the noise depend on V, and there is a kernel, a memory kernel, a bit more complicated. But you see that in principle, this Ornstein-Uhlenbeck, you can refine and have a very, very precise description of cell motion. Okay. So now that I've reminded you a little bit what cell motion is, we'll go to collective cell motion. And the first thing that was described a long time ago, actually in 21 by Leo Loeb, is it's a very important process. It's called contact inhibition. And this was really studied in great detail by Abercrombie and his collaborator in the 50s. So what is cell inhibition, contact inhibition of locomotion is that the fact that two cells don't like to move on each other. So when they come together, like here, they make contact and then they stop making protrusion toward the other cell. So there is protrusion inhibition here, and then they change direction. Okay? So it's a very simple process, but it's very important. Oops, sorry. Okay. So you'll see that in real. So this is a movie that I took for Roberto Meyer. So you have these two cells that are coming together, they make cell contact, and then they just remove. So the importance of that is that if you have dense cells, they will stop moving. So the cells that are confluent stop moving. And also, this cell will not crawl on each other. They will just stay as a monolayer. Okay. So now, what are the models for collective cell motion? So there are different types of model. One type is, as you heard in the previous lecture by Hugues, are just continuous media models. So you write this kind of hydrodynamic equation for the velocity with different terms, usually based on symmetry argument. And these are actually nice because for continuous equation, there are lots of tools to analyze this equation. As Hugues was saying, you can look at the stability, etc. And I suppose that Jean-François described this, how do you I mean, what these symmetry arguments are and how you can write some of these equations on symmetry equation. So these are nice, but these are usually, I mean, not usually, they are valid on a scale that is larger than the scale of the cell. So if you want to see things that are really on the scale of the cell, like this, for example, these fingers, these are not really good description because the elementary element plays a quite important role. So there is really some reason to look at cell-based model. Also because if you look at cell-based model, you have a more direct relation between cell dynamics, some phenomenon at the cell scale that you can include more easily in cell-based model. You can also look at a little bit more easily at fluctuation, stochastic phenomena. And so many people have looked at this kind of model, and there are different levels of model. The simplest one is just the V-check model that Hugues described. But this V-check model, if you want to make contact with experiment, is a bit too simplified because you see that it has, for example, a single velocity, and if you want to describe what is the velocity of the cell, clearly it has not a single velocity. So you want a simple model, but a bit more complicated than this V-check model. And so some continuous model. And so in principle, you like also to use what is known for the single cell. I mean, this very simple description. So this has motivated many people to use a model of this kind. So while just this would be like the single cell in some sense, where you have the velocity, you have the noise, and then you have supplementary terms. So one would be interaction with other cell, and also in this collective cell motion, the motion is organized on a larger scale. So a simple way to do that is to add a coordination term. It was, one can say it's a V-check term, but you can also say it's just a Navier-Stokes term. I mean, it's just viscosity in some sense. You couple one cell would like to have the same velocity as its neighbor. So it's exactly like viscosity. Okay? So this is a simple term, but at the clearly this is very phenomenological. As the single cell model, we didn't describe in detail how this alpha is coming and the noise either. So this is really phenomenological model. And so we try to compare a model like this with data from Pascal. So in our version, we put also that the noise is Ornstein-Uhlenbeck, so that we don't have white noise. We don't have things that change immediately. All force have some persistence. And in this kind of model, since you describe cell as point, you don't want them to overlap. And so there is a repulsion between this point. And you can also put some attraction if they are a bit separated and not too far. So there is an intermediate range attraction from a point with its neighbor. So one can try to see if this model accounts for the data in some sense. So we compare this with what Pascal has measured. So I just show you, so these are the velocity field in the experiment and in the model. And so the parameter of the model have been adjusted so that things look similar, of course. So it looks actually similar. And if one compares, say, the velocity distribution in the model, these are the colored dots, and in the model, this dashed line, you see that it fits quite well. Here it's the velocity correlation at equal time. So again, it's quite similar. The velocity correlation at 2 times, it decays basically the same in the model and in the experiment. So this simple model is able to describe, at least in a statistical fashion, what you see in the experiment. You see also given that you have put a potential that prevents cell to be close enough. So if you look at the structure function, the probability that if you have a cell at 0, you have another cell at a certain distance in the experiment and in the model. So you see that, okay, you have excluded this region by the potential, and then you have this kind of thing, and then the distance, you basically you get the same quantity in the model. So a model like this, if you adjust parameter, is able to reproduce the data that you have. Okay, and it's also possible given that, okay, so here what is the complication? Here the complication is that you have a coupling, the model, this part is purely linear, as you see, but now you have a coupling with cell position that now is nonlinear. So one way, very easy way to just try to understand what this model contains is to put, say, let's say that this particle are on an hexagonal lattice. So all this force will balance, and this will be 0. And we can forget that V change the position of the cell. So basically, we can just solve this equation for the velocity, which is a linear equation. So if you do that, you can get, it's just a linear equation, so you can get the solution by Fourier transform of this equation, and then you can compare with what you obtain. And actually, it's a quite good fit of the correlation in space, slightly less of the correlation in time. So it means that motion reduce a bit the correlation in time, the autocorrelation of the velocity, the fact that cell moves reduce a bit the correlation. But basically, with this approximation, you see, well, you can see how things depend on different parameters. Okay, so what is the lesson here? So the lesson is just that the motion of cell in this kind of experiment is compatible with a very simple model. That the velocity, that's data, the velocity are correlated about, say, about 10 cells in size. So it's not that you lose correlation. There is a quite large correlation. And this, of course, we reproduce by putting this alignment term. And so there is a velocity alignment, but there is no indication of nonlinear term in the velocity. So no indication of preferred velocity. Here you get a preferred velocity because you have a strength of the noise. Okay, so it's a nice fit to the data, but why is it interesting? Okay, so a model like this is interesting only if it helps to understand something else. Otherwise, just a fit to the data. Okay, so I just will describe what we have tried to do, and so it's to apply it to global fluctuation in confined cell assembly and guidance by leader cell. Okay, so this, I will describe this experiment, again done in Pascal group. So before I was showing you experiment where the cell were invading free space. So now this is, the setup is a bit different. The cell are put here on adhesive surface, but the adhesive surface is just a small disk, and here it's non-adhesive. So this cell will divide and multiply in this area, but will be confined in this area. Okay, so I'll try to show you the movie. So you see that you have motion and then cell multiply. You have more and more cell, and at some point you'll see that the motion will cease. Okay, then it's not so moving. It's moving less and less. So I'll show you the data for that. So the fact that it's moving less and less is a reflection of this contact inhibition of locomotion. Okay. So this is the experiment. So you see that the velocity, so this is time, cell multiply, so the velocity decrease. And if you look, say, at the division time, there is also something that is called contact inhibition of proliferation. So if you are more dense, cells tend to divide less. So if you look at the cell area, it diminishes as time proceeds, and then the doubling time of cell is increasing. And so here it's in time, okay? So you see that it grows with time. So you have this division of cell. Okay, so what you see here in the velocity, so that's the mean velocity, you seem to have large fluctuation, at least in this intermediate region where cells have become confluent, but they are not yet very dense, so they can move, so you have fluctuation. So what do you see here? So you see something that is interesting. So Pascal and his group decided to analyze this motion of cell by decomposing it into orthoradial motion and radial motion. So I'll describe you, first of all, what is the orthoradial one, so like this. So what you see here is this u theta, the mean orthoradial velocity. You see that it goes up and down, so you have reversal. So the thing goes to rotate in one direction and then stochastically rotate in the other direction. And so these are the profile of this orthoradial velocity for different, yes?
From your movie in the previous slide,
yes?
it looks like, it may be a dumb question. From your movie, it looks like that the cells are dying rather than it's
the cells are what?
The cells are dying.
No.
Rather than the slowing down of motion, is it like that?
No, I don't think they are dying. But maybe what you see is that at some point they are, I mean, the thing becomes three-dimensional at some point, but I don't think.
It looked to me like as if like they're rounding off.
Okay, so you want to answer, Pascal? They are rounding. They become maybe.
Divide. So sometimes you see white spots that is dividing. And at the end of the sequence that Vincent showed, some of them get extruded in the third dimension. So the result, I mean, just the result of cell division in a too dense environment. So
they don't stay in the plate. They go out, but they don't die, I think. I mean, maybe some of them die, but not a lot. Okay. But in any case, I won't go into this later period. I will stay when they are on a 2D surface. Okay. So the later period is also interesting. You have 3D structure, but we won't discuss that. So in any case, so here what you see is this orthoradial velocity as a function of radius, and you see that it changes sign. Okay, so it's go from 5 micrometer per hour to -5. Okay, so there are rotation, and there is something that is really puzzling for physicists is that if you look at the statistics of clockwise and counterclockwise rotation, it's different. So okay, so this is a parity breaking, but at a macroscopic scale. Okay, so that's interesting. So I was very surprised from that. Pascal told me, okay, no, it's well known, and in fact it is. Well, I mean, it's well known, but for those people who know the recent literature. So here, for instance, you see it on this same kind of setting when just this micro-pattern mammalian cell. So you just look at the image and people have quantified this thing. And in fact, you see that there is some chiral pattern here. Okay, so this is not symmetric. And in fact, you have more cell going in one direction than in the other. So there is something that I don't think at the moment is very well understood, is that, okay, the basic molecules are clearly chiral, but this is transferred at the macroscopic scale. And so, okay, so in any case, it's a fact. Interesting fact. Okay, so that was for the orthoradial velocity. And now, if you look at the radial thing, then you see more periodic oscillation. You see here, this is the average radial velocity. And here, if you look at the profile of this velocity, it's bigger in the center of, I mean, that's the center of the disk, that's the perimeter, so it's bigger at half the radius in the center here. You see it here, and you see that it oscillates somehow with a certain typical frequency, and if you plot this frequency as a function of the radius, it grows with the radius. So the period grows with the period. The oscillations are slower for larger disk. Okay? So that's the phenomenology that is seen in this experiment. So now you can ask, this is biology, so where is this coming from? So it could be that there are new mechanisms that are induced by confinement, that there are waves that are coming from induced by the boundary, things like that. So the one way to look at it that we choose is just to, so we have this model that has been fit to the motion of this cell, so it's quite easy to put this model in this particular geometry by putting an external repulsive potential. So we just took this persistent Brownian Walker and confined them to a disk. Okay? So we can see at least what is emerging from the basic interplay between persistence and noise. Okay, so that's what we did. And when we did that, actually, we find something that is very similar to the experiment. So we can look at this reversal in the orthoradial direction, and here you see the reversal statistics in the model. So it's really very Poissonian, actually. Maybe less than here, it's a bit different, but I mean, it's not complete. I mean, it looks a bit the same. And of course, there is no difference in the simulation between the two rotations direction, because we didn't put anything that breaks chirality. And if you look at the profile, well, they look also a bit the same for this orthoradial experiment. And again, if we look at the radial displacement, this is the simulation. So there is just the model of the previous that was fit to this large scale motion. You see that actually it's very similar to what you see in the experiment. So basically, this is the model, this is the experiment, and they look very similar. And they look even more similar if you go so these are just the power spectrum of this mean velocity in the radial direction. So you see that they are not very sharp. I mean, these are like broad oscillation, if you want. There is a peak, a preferred frequency based in the experiment and in the model. And if you plot them on top of each other, actually you have no adjustable parameter. You just see that it looks quite good. So and of course, so this tends to say that, okay, there is no, what you see in the experiment is actually just a product of the fact that these cells are persistent. They tend to have a correlation length that is the size of the disk, and that there is noise. And in fact, you can test the role of cell coordination, both in the experiment and in the model. So in the experiment, you can put things that suppress cell coordination, and you can also test the role of cell division. So if you put this mitomycin, you're supposed to see that it's like the control in some sense. So that cell division is not really important for this oscillation, but that if you put things that suppress coordination, then the peak is not so large. And of course, you can do that in the model much more easily by just suppressing coordination, this beta term in the model, and you see that also the amplitude of this thing is very much reduced, the amplitude of this oscillation, and the frequency is shifted. Okay, so that just to see that the model contains that. And now you can ask, where does this come from, actually? So in the in the model you can do things that are there are things that are easier to do in the model than in the experiment, and one thing that you can do very easily in the model is to put the model cell in one dimension. Say, if you look at one dimension, we'll say that, okay, the orthoradial thing, the orthoradial motion is just, you see that it was bigger around the periphery of the disk, so we'll just take a line with periodic boundary condition and we can look at that. So these are simulations in one dimension, and you see that they are reversal exactly like in 2D. So if I look at the line of this walker with periodic boundary condition, the velocity will go on one and the other. So where is this coming from? So now this is rather easy, because if you are in one dimension, we can sum all these equations, and basically for all the interaction term will vanish 2 by 2. And then we'll have just that the mean velocity is an Ornstein-Uhlenbeck process. Okay? So this is exact. There is no approximation here. Not really Ornstein-Uhlenbeck, because the noise itself is colored here, but at least we have these two equations. And then, so the mean time between reversals, so here we have intervals where there's velocity reverse. So the mean time for a process like this is something that one can compute. This was done by Rice in '54 for general Gaussian motion. But the statistics of zero, so here you have a zero crossing, so the velocity is going in one direction, then changing direction. So if you want to look at the period, you want to look at the statistics of the interval between the zero crossing. So this is a quantity that actually even for Gaussian process, nobody knows how to compute exactly, but there are approximate way of computing this distribution. So one approximate way, so something that is easy to compute here is the correlation function of the Gaussian process. And so what you can compute is, if I draw this interval independently but with a given probability distribution, what is the probability distribution that would reproduce the correlation function? So you can do that. So you can do, and that's an approximate way, just neglecting correlation between the length of two intervals to get an approximate formula that actually works quite well. So this is what would give this approximate thing. And you see that you can understand. So what it says is that the statistics of this reversal is actually a random process, but the correlation is coming from the fact that this is persistent. Okay? And you can also look at the oscillation in one dimension, and again you find something that is very similar to 2D. If you so now for the oscillation, what you put is a fixed boundary condition, so which would a little bit mimic the radial direction. And now you have again things that depends on the size of your 1D box. And this, again, in one dimension, there is a simple approximation, is just to say that these cells, basically the only thing that one can compute are linear thing and then correction to that. So just saying that the interaction is harmonic. So you have just your cells and you put, so here I've written it in the continuum just to be simple. So basically you have some harmonic interaction between the cells. This was the alignment term. And if you do these two things, again, now it's linear, you can solve it and you can, it agrees with what you see basically. So the image that one obtains is that this stochastic reversal is just a Gaussian process. So there is no complicated thing as what one could have imagined, like a nucleation or things like that. And for the fluctuation, the oscillation is more like that you are somehow filtering the noise that each cell injects into the process with some filter that is made from the interaction between cells. So that's actually the picture that we have. Okay, so that was for this confined experiment. And then one other way we try to look at this model is looking back at the progression of this interface. So I showed you that this was driven by leader cell, etc. So can we look a little bit more into that? So if you look back at the experiment, what you see, one thing that is seen is that there is a crossover. It starts, so if you look at the mean advance of this interface, it starts quadratically and then linear. And then, of course, it stops when it encounters the other boundary. Okay, so that's data. So that's one kind of thing, for instance, that one would like to understand better. And also, what are these fingers? So this has this kind of the finger formation. So this digitated interface has attracted the interest of several people. So there are different hypotheses. One is that it's really an instability, so it could be mediated by diffusion and chemotaxis. So that was proposed by several people. And there is also a simple model by Nirgov in Israel and Pascal saying, okay, maybe what is happening is that, okay, the interface is, if the interface is curved, for some reason, cells that are at the place where the interface is curved can go faster. So let's suppose that the velocity of cell at the interface is proportional to the curvature of the interface. So if I have a curved interface, maybe I should draw that. So this part will go faster than this one. So the velocity will be, say, proportional to the curvature of the interface. So if you say that, this is an old model for crystal growth. Actually, it's called the geometric model for crystal growth. At a later time, what you would have is that this thing is like this. And this will go even faster than this one. So you see that you have an instability automatically if you say that the velocity is proportional to curvature. So that's one thing. But OK, so that's what is creating this leader cell at initial stage is not very clear. There are different possibilities. But so what we try to address is a kind of different problem. Given that you have created this leader cell, what can they drive the rest of the assembly? So these leader cells, they have a different phenotype than the other cells. So they have the other cell, they are very directional, so they move really in straight lines. So here it's the displacement, say, parallel, perpendicular. And you see that they move at a rather constant velocity. And you see here the distribution of velocity. So they are faster than the mean velocity in the bulk and very directional. So the question that you can ask is, OK, if I put a faster cell in a bulk of this other cell, can I produce things that resemble what I see? So yes?
Sorry, how many leader cells do you have? Is it all across the boundary? Or?
So no. So OK, so that will come in the next slide. So this cell seems to appear a bit randomly along the boundary. So you have few of them. I mean, but I'll show you the statistics.
What happens if one of them is damaged?
What?
If one of them is damaged,
Is damaged? If one of them is damaged, so I'm not sure this has been done. So what you can do is cut the interface. So then, if it is not longer linked to the interface, is a bit lost. If you are a leader without followers, you are not happy. So they just go a bit mad. And then when they they again come in contact with the bulk of the thing, they are happy again and start. And also, as I will show you, you can induce the formation, probably induce the formation by rupturing some actin cable that goes along the interface. But we'll come to that. Okay, so the question is exactly that. So I will put, suppose I put one, can it entrain the other one? And also, what does it, what should I do to produce shapes that resemble what I see? And of course, if I understand this, maybe if I put a few of them, I can understand what is this quadratic regime. So that's the idea. Okay, so now if you try to do that in the computer, this is not very easy. So if you put a faster cell, okay, then what happens? It goes fast alone. It just goes away because the other cell cannot go as fast. Okay, so you say maybe, okay, let's put a very strong interaction between this fast cell and the followers. Then, okay, it goes with 2 followers, and that's it. So actually in the model, it's quite difficult to entrain a lot of cells. So you can say, okay, maybe there is, so as I will show you, there is an actin cable along the interface. So you can say maybe, okay, this actin cable is very strong. So you put this fast cell, and then you have like a pinched string, which doesn't resemble at all what you see. So what we convince ourselves, or at least in this computer experiment, the idea was that if the follower could not attain this leader cell velocity on their own, it was really difficult to get this. So then if you come back to, so what are the mechanisms that can do that? So maybe there is pressure. So, okay, but this doesn't seem very plausible. But on the other hand, there is this contact inhibition. So when cells become less dense, they tend to acquire larger velocity. So actually, if you say that the velocity of the cell is inversely proportional to the density, which you can see by correlating the local velocity with the density, then local cell, cell in the bulk can acquire large velocity. Yes?
Sorry, do you observe any cell bladding, for example, which is also linked to cell movement, for example?
So cell, so here what you see that all these cells, even in the bulk, they have lamellipodium. So it's very obvious for the leader cell, but even the cell in the epithelium, they have what people have called cryptic lamellipodium, so they move also. Okay, so they are all polarized, they all move. Okay, it's not that they are not moving in the back, they are all moving. And so what is happening here, what we propose at least is that, okay, if you have one leader cell, then the density decreases, and then the follower cell also can go flatter on the substrate and move faster. So even in the bulk, the velocity can change with the density. So that's an important ingredient. And the second one, okay, so the second one is that if you want to have this leaders, these fingers, is that elsewhere, when you don't have a leader cell, the cell should have some difficulty in invading the free surface. So that's the 2 ingredients we found necessary. And so the idea is that here we have the interface, there is an actin cable, and so this may prevent the lamellipodium of this cell to develop in this axis, and so that they would have difficulty in moving in this way. So one experiment that Pascal did is to cut this actin cable with this with a laser, and then what you see is that these cells with a high probability tend to become, to develop a lamellipodium and to become a leader cell. So the idea is that, okay, you have this leader cell. When the leader cell goes on the surface, then the other one are happy to follow it and move fast. But here, it's difficult to invade, okay? So what we did in the model is to say that, okay, there is some repulsion here, everywhere in this free surface for this normal cell, but that the leader cell can remove this repulsion, and so that the other cell, and put a density-dependent noise. Okay.
Is it really an actin cable, or is it just, because my understanding was that the actin was forming a network, yes. The network conformation at the...
So there is the actin for the lamellipodium, which is branch actin. Okay. But then there is also a cable of actin that is actually going from cell to cell and just interrupted in the leader cell.
Okay, so it spans multiple...
Yes, yes. So I think you can see it more here in the previous slide. So here you're supposed to see it actually, this cable, okay? That's lined the interface. And that's actually ruptured by the laser.
Sorry, can you do multiple cuts and get multiple...
What? Can
you do multiple cuts at different places and get multiple leaders and get branching?
You can if you go to Pascal Lab, I think. I didn't do any of this experiment myself, so... Okay, so this is the model, so what you get in the shape of the fingers, and so this is this repulsion that is destroyed, and then, okay, you get things that resemble a bit these fingers. So the ingredient that we came up with is that this normal cell can move on their own fast if the density is small enough, that they do not invade free surface easily, and basically also we put that, okay, if the leader cell goes, nobody is following it, then it stops till there are again some motion, so it coordinates its motion with the follower. Okay, with that, we can reproduce things, and then we come to this question of the global motion of the interface. So basically what we say is we put a cell randomly in the interface with this velocity that was measured, and here you see that there is a number of leaders that were appearing, so I think that's per millimeter of interface, and then they stop basically after 20 hours, which also correspond to the division time. You have much less leader production. So this is basically what we put, that we put the creation of leader up to here. And if you do that, you obtain things that, okay, is a bit resembling the experiment, okay? So you see this invasion with these leaders. Okay, so, and now in the model, you have also this quadratic and then linear progression, okay? And okay, you have velocity profile here. The velocity, I mean, the things is wider as time evolved. You have more and more cell, both in the experiment, which is dashed, and the simulation where you have more and more leader. More and more cell moving fast. So where is this quadratic thing coming from? So this is rather easy actually. So this quadratic thing is that, okay, if you don't have leader, the interface is moving with a slow velocity. And when you have a leader, then the cell move, you have a chunk of interface that move at a fast velocity. So if you had a fixed number of this chunk, then it would move at a linear velocity, which is given by the number of slow and fast part of the interface, an average. But what is happening is that this number is increasing linearly with time, because since this leader cell appear as a Poisson process randomly, you have, as time evolves, you have more and more rapidly moving chunk, and this more and more is linear in time. So if, yes?
Do they also disappear randomly?
I don't think they disappear. Do the leader cell disappear? I'm not sure. This maybe happen, but very infrequently, actually.
So once you're a leader, you're
Yes, that's actually the. In any case, in the model, this is the what we see. So basically, if you have these two ingredients that a leader cell is just is following by a chunk of interface of a certain width, and that you have more and more of this thing linearly in time, you get this t-square increase of the average motion. So that's a simple explanation. Okay. So basically, I think I can conclude here. So you see that even very simple modeling can account for many features of the collective dynamics of the cell in vitro. And the interest of simple model is to see what follows directly from what has been included and what requires a supplementary explanation. So then you can ask what else? Okay, so what else? So clearly there are several, yes? Two? Okay, we come to that, yes.
So he also observed that there's a migration.
So I think that's what was written here. But
there are two populations of cells that merge together, and he also observed fingers.
Yes, yes, so we'll come. I'm just going to discuss that. So okay, so 1 minute thing. So okay, so there are different things that need to be clarified clearly. So one thing that would be nice that, okay, we have, in this simple model in general, there are just phenomenological parameters. So people want to link them to some molecular pathways, to some molecular modules. So this has already been started by different groups doing RNAi and looking, so you knock out one gene and you see what it affects. So at the moment, people have done that more on a qualitative way, but this kind of simple model, I think, is a way to do that more quantitatively. You can ask, in principle, when you knock out something, which parameter does it affect, and quantitatively. So this would be a way to do this module a bit more quantitatively. So I think that's and also to get some more understanding, for example, of this alignment parameter, where does it come from? So that's I think, a nice project that will be pursued in different groups in the future. And of course, you have these simple models. I mean, I've described one, but there are several that are exactly of the same type. And so the interest is, of course, to see if they can describe more complex phenomena. So already, as you mentioned, there are nice cases in vivo where this has been attempted. So one is a mesendoderm progenitor. So this was in the group of Carl Philipp Eisenberg. So he has a motion of mesendoderm progenitor on the zebrafish. So the model is basically the same, except that there is a gradient, so that the cell move even without collective, a single cell would move in one direction guided by a gradient. But if you put collective interaction, exactly like I put here, I mean, they use a model that is very similar, then the collective will move more efficiently toward the gradient. So that's one in vivo case that has been studied. And as you were pointed out, so there has been a lot of work by Roberto Mayer on neural crest. So the basic model of neural crest is the same, but there are also more complex ingredients. so what they do, so these neural crests, they are more diffuse, but so they put contact inhibition so that the cells scatter. So that would just give scattering of cells. So actually what they characterize is a local chemoattraction between these cells. So this cell release a molecule that is a complement molecule, C3, that makes local attraction between them. So they attract locally. So there is cohesion that is mediated by a chemotactic molecule. And then even more, so the idea that in the simplest model, they have this contact inhibition, which say that the front is moving faster because it's undergoing less contact inhibition, very similarly to what we postulate for the fingers. But there is the adhesion there is mediated by this local chemotactic thing between cells. And then these neural crest cells are moving on placode cell. So that in the next step, so basically the placode cell, there is a kind of chase, and I don't remember how they call it, chase and run. So there is one they follow and they just chase and run because of placode and neural crest cell. And in this case, there is also, as you were saying, some kind of digitated appearance of the interface. I don't think they have really studied what is, so that's actually something interesting. I'm not sure if they understand precisely how comes these.
They don't have leader cells, or?
Well, it's what they call leader cells are the ones that are in the front that do not undergo contact inhibition. So you're right, but this is more coming. If it's coming, it's coming. I mean, there is also, so there the chemoattraction can play a role because it had been well characterized, this C3 thing. So there are chemoattractant-induced instability. And then there is this complicated thing with the placode cell. So I think this needs to be studied to see where this instability is coming from. Okay. Okay, and of course, I think there is need to study how the assembly, if you have one model, even simple of the cell, so that was what Hugues was talking about. Here, if you give one element, if you put them together, what will be the effective description of that? And I think there is also some, so of course you can do that in the bulk, as Hugues was discussing, but I think there is also an interesting kind of problem which typically we had in this confined cell assembly, which is more mesoscopic if you want statistical mechanics, if you have a few hundred entities, so not a very large number, so that you have fluctuation on the global scale, which has not been too much studied in statistical mechanics. So what are more this fluctuating hydrodynamics of a few hundred entities. Okay. So time for conclusion and lunch. So I just took this conclusion from Michael Abercrombie in this Cronian lecture. So Michael Abercrombie is the guy who pushed a lot this social behavior of cells starting in '53. And so he has this Cronian lecture just before he died. And so that's the conclusion. So he says crawling is one of the most important cell activities in the animal kingdom, important in its generality and its function, okay? And then he says, of course, at this stage, no general picture can be satisfactory, but to generate dissatisfaction is perhaps the main purpose of a general picture, and I'm sure I've succeeded in that. So I think I can take this last sentence for my conclusion. Okay. And then, of course, again, the picture of everybody, including Pascal, who is here. Thank you.
that. I feel very embarrassed to ask a question that below my own picture.
An interactive modeling was you keep derivation always of velocity. So do you assume to present some inertia in the system?
So okay, so that's a question even for the standard picture. I mean, it's not our thing. It's coming back to the 20s. Okay, what is this inertial term? So I think it's more a memory term if you think of it. So basically you obtain that. So what it says is that the velocity at a given time does not depend on just the force at this time, but on the force with some exponential weight on all the force that you have perceived in a certain time, if you integrate just. So actually you obtain that maybe more naturally in models where you have two ingredients. You have the velocity and the polarity. And if you say that the polarity wants to align with a certain time scale to the velocity, then again you get this memory effect. So this memory effect you can get for if you have other variables that give the velocity, and I mean that have some dynamics that is not instantaneous, but that give the velocity. So that's and actually, if you look at this data from Flufberg et al., I think in any case, in a phenomenological way, it's quite well supported. So that's not that it's completely crazy. Although for physicists, you say, okay, this is inertia, so it's crazy, it's a low Reynolds number thing, but I think this is not the case. I mean, it's more memory that you have, because the cell is some complicated thing.
I was wondering if your model could give you access to some global mechanical properties like shear moduli or something like that.
Okay, so it's something we did. I mean, it was Pascal has measured local force if you have this thing walking on substrate, or you can. We didn't do. Maybe we should do it. Yes, no, it's an interesting thing. Yes.
What's the sort of length scale for the attractive interaction that you put in?
So we just said it's the neighbor one.
What?
Neighbor.
Okay.
So the neighbor.
Local.
Is what? It's completely local. Yes. It's a local thing. So, but even we say, okay, so there is here we have a cutoff. If the local is far, it doesn't feel it. So it's attractive interaction if the two cells are not farther than their size, basically. It's just because we describe them as point. So we have a repulsion so that they don't overlap, and then up to a certain distance, which is basically two cell size, they attract, and then if they don't see each other.
So during this lecture, this course, we had many lectures on inference techniques in which, for example.
Okay, so yeah.
So I was wondering. In your last slide, you were talking about fluctuating hydrodynamics for this kind of models. Do you think that it could be possible to apply this inference technique to build?
So for example, this I showed you this work by Flufberg because it's exactly what they did. I mean, they just look at the data, and then they measure root mean square velocity, the distribution of acceleration, and then they build their model from that. Okay, so that's, I think it's a very nice procedure. Here we didn't do it, probably in part because we were lazy, so what would imply? It would imply that you have to segment all these movies and then compute the average with fixed, say this. I mean, you have to process a lot of data to do that. In principle, it's a worthy enterprise, but we took just a simpler way, just try to fit a simple model. But you're right that if we want some more precise description, a little bit like you did with fish. I mean, you can follow the two fishes, and then you build really your model from. So you could do that. It's kind of. Yes, it's still worth doing it, certainly.
Coming back to this clockwise,
yes,
counterclockwise,
yes,
trading observation. So obviously, I guess the obvious first candidate might be the actin filaments that are actually strongly chiral. So I was wondering whether you could try to build some quantitative arguments.
So I think it actually is more subtle than that. I mean, my own view is actually there is very interesting work on nano objects. So if you take nano objects that are not chiral, then in certain conditions you can have spontaneous chirality breakings simply for entropic reason, that if you put them, that was an old argument. I mean, this is basically going back to Onsager for the nematic transition, that you have more entropy if you align things. So in some cases, if you take rhomboids, and there are experiments on that, they just take I mean, of course, the two chirality play equal role, but you would have spontaneous breaking of chirality. So now my feeling is more than here what we see is that, okay, the basic phenomena would be this spontaneous breaking of chirality, and that you have a small term that breaks it. So what I'm saying, so that you have also a small field that bias the two things. I think you need some clear amplification. So it's pure speculation, but I think if you go from...
I think you don't actually...
Okay, maybe
well,
not.
I think what it hints to is that this is not kind of chirality and continuous symmetry breaking, because it hints that you have some strong chirality underlying, but you actually have in the cells, for instance,
okay,
actin filaments are definitely, you know, one chirality and not the other.
So it's not, I mean, maybe I should, okay, so I don't think it's immediately related to chirality of actin, because if you look in this paper, if you look at this paper in PNAS, they took different kind of cells, and different kind of cell break chirality in different direction. So it's not directly, so maybe they use actin differently, but I mean, these are a little bit similar kind of cell, and some of them prefer to go in some direction, and some of them in some other direction. So it's not straightforward. I think it's not completely straightforward. So, but okay, so in any case, it's not, I think it's an interesting thing, but I don't think there is any good explanation at the moment. That's why I put the slide. Yes?
Thank you.
Okay.